we are going to talk about Mendelian genetics. So, Mendel was a monk, and he had a garden, and that's what he did. He tended to his garden, and in his garden were some pea plants. And some of them were purple, and some of them were white. And Mendel wondered why that was, and he decided to do some experiments. So he took lots and lots of purple plants and crossed them again and again and again and again until he was sure that whatever was going on inside them, he didn't know it was DNA. Anything that they were carrying said, "Make me purple," and nothing else. And we call that true breeding, or purebred. And so he took a purebred purple plant, and he took a purebred white plant as well. He did the same thing with the white plants, and he crossed them. And he called this generation P for parent. And when he crossed these two plants, he saw that he only got one type of plant in the offspring, and this was purple. And Mendel thought that was strange, because where did the white plant attribute go? So he decided to look into this further. And before he looked into it further, he named this generation of new plants, he named them F1, because he knew he was going to do more of these generations, so we wanted F2 and F3 in the future. And F stands for filial, which is Latin for offspring. So these plants got crossed with themselves. And when they got crossed with themselves, I'm going to draw another arrow here. But anyways, when they got crossed with themselves, they ended up making a fixed ratio of purple and white plants. And this ratio is 3 purple to 1 white. And Mendel decided that this was because the heritable information, we now know that it's genes, could be dominant or recessive. And dominant meant that if you pass on one version of it, then that version will prevail. It'll show. But recessive means that if you pass on a version of this gene, or an allele is a version of a gene. So if you pass on a recessive allele, then if you also get a dominant one, the recessive one is going to be masked. So this made sense to Mendel, because if these parents each had one allele from each of their parents, and they were purebred, then the purple one had 2 dominant alleles, and the white one had 2 recessive alleles. So we like to symbolize dominant alleles with capital letters and recessive alleles with lowercase letters. So if we call this P for purple, or P or plant, or whatever we're talking about, this has 2 capital P's, and this has 2 lowercase p's. So every single one of this F1 generation got 1 capital P from the plant on the left, the purple one, and 1 lowercase p from the plant on the right. So both of these guys are what we call heterozygotes. So they have one of each type of allele. And when we say heterozygote, hetero means different, and zygote is like an offspring in its very earliest stage. So heterozygotes have 2 different alleles. The guys at the top, we call these 2 homozygotes. So those are homozygotes because they each have a set of the same alleles, homo meaning same. And then this F2, the reason why it was split up like that is because of probability, and we can draw this handy little device called a Punnett square in order to help us see that probability. So each of these F1s could pass on one of their alleles, because they're each going to make gametes where they have half of their genetic information in that cell. So they're going to be passing on one allele of the 2 of them that they have. So the one on the left could pass along either a big P or a little p. The one on the right could pass along either a big P or a little p also. So if we look at the different combinations that we can get from this, we can get two big P's, or homozygous dominant. We can get one of each. We can also get one of each here. So these are the heterozygotes. And we can get homozygous recessive over here. And these different combinations of alleles, we call these the genotype. But what we're looking at when we're looking at purple versus white is what we call the phenotype. And the phenotype is however that trait actually gets displayed. So how you look, or how you behave, or how you function inside, that's your phenotype. But your genotype is which alleles that you have in your genes. So in the phenotypic ratio, we have 3 that are purple, because in all of these they have a big P, and that's the dominant allele that's going to prevail, and that is purple. And then the one that did not get a purple allele is going to be white, because it didn't get a purple allele. So that's where we get this 3-to-1 ratio. If we had different parents for this F2 generation, we would have a different ratio. It's just 3-to-1 because that's what happens when you cross 2 heterozygous individuals. So let's say that we were crossing a homozygous dominant purple plant with a heterozygote. So if we had a homozygous dominant, that's 2 capital P's. So this purple plant that is homozygous dominant can only pass along a capital P, and it can only pass along one of them. So we can either write 2, I wrote 2, or you can simplify it down and only write 1. So we're crossing it with a heterozygote, so that's a big P, and this is a little p. I'm going to try to make it very little, because it's hard to tell when they're not next to each other. And this one also looks purple, but its genotype is different. So let's see what happens. Well, in this combination, where they both give a big P, the genotype of the offspring is homozygous dominant. If they also both give a big P, because that's kind of all that the top one can do, then it's also going to be homozygous dominant, and the other 2 genotypes are going to be heterozygous, because the top one is giving a big P and the side one is giving a little p. Right? So this is a 1-to-1 genotypic ratio of homozygous dominant to heterozygous, and it's also a 1-to-1 phenotypic. Wait, no, it's not a 1-to-1 phenotypic ratio, because all these guys, their phenotype is going to be purple. So we are only getting one phenotype out of this. So that's how you predict what happens when you pass down genes. So we can do this in people too. So there's a thing called a widow's peak. It's when your hair, instead of going like this is a straight hairline. You can have a widow's peak, which can look like this. And the widow's peak is a dominant trait, and it works by this complete dominance thing that we've been talking about. So if I'm talking about 2 humans, and one of them has a widow's peak, but had a parent that didn't have one, and this person is going to be crossed with another person who does not have a widow's peak. So the one who has one, they either have to have 2 capitals, or they have to have, a capital and a lowercase. So they either have to be homozygous dominant or heterozygous, right? But since we know that one of their parents didn't display this trait, this dominant trait, that parent must have been homozygous recessive, or little w, right? So in that case, that parent had to pass down the little w allele because they didn't have a choice, right? And so this person is going to have to be a heterozygote because of that. So we're going to separate these out. We can either give big W or we can give a little w. Meanwhile, the other parent is homozygous recessive because they do not have the trait, and that's the only genotype that you can have if you don't have a dominant trait. So this person does not have a widow's peak, can only pass on not widow's peak alleles. So what's this going to reveal? We're going to get here heterozygote, here homozygous recessive, here another heterozygote, and here another homozygous recessive. So this is also, like the other one, half and half genotypic ratio. But unlike the other one, this is a half and half phenotypic ratio as well. Or a 1-to-1, sorry. Half and half doesn't make sense for a ratio. Anyways, it turns out that Mendel wasn't right about this working for all the different ways to inherit things. There's also something called incomplete dominance. So in incomplete dominance, you can see the heterozygotes have a different phenotype than either of the homozygous phenotypes. So you can see this in snapdragon plants, actually. So in snapdragon plants, the homozygous dominant for color is red. So I'm going to call this allele R for red. So this one has this phenotype. And the homozygous recessive is white. But the heterozygote is actually going to end up being pink. So this is what incomplete dominance does. Heterozygote, and I don't have pink, I'm going to use red, and I'm going to try to shade it like a little bit less darkly. So that's pink. Pretend it's pink. Okay, so in this one, if we were to cross, say, a red snapdragon with a pink one, so these two guys, what would we get? Well, let's make our Punnett square. The red one can either give a big R or a big R, and I'm going to try just making one column for this then, because it can only give a big R, and this one can give big R or little r. So you can get either a homozygous dominant, which is going to be red, or you can get a heterozygote, which is going to be pink. So it's going to be half and half. There's also something known as codominance. And in codominance, basically you can have more than one dominant allele and they can both show up at the same time. And the classic example of this is ABO blood type. So we have red blood cells floating around in our blood, and they have what is called cell surface antigens. And what an antigen is just like a little molecule that helps us determine whether this is us or some evil bacterium that's trying to infect us. So there's a couple different kinds of molecules that we can have on here. One of them is called the A antigen. So this will be A. One of them is called B. And then if you have O blood type, you have neither one of them. So this person has AB blood type. If you have A blood type, you only have the A antigen on your cells. And if you have B blood type, then I'm drawing these wrong. These are like sickle cells. Oh my gosh. Okay, I need to draw them as like these. They look more like this when you're a healthy person. Okay, so these are healthy people. Anyways, so if you have B blood type, you're just going to have the B antigen. And then if you have type O, then you're going to have nothing. And the way this works is there's a gene and there's an allele that makes A, there's an allele that makes the B antigen, and there's an allele that doesn't make anything. And so we call these alleles IA, IB, and lowercase I. So the IA allele makes A, the IB allele makes the B antigen, and the lowercase I does not make anything. So if you are type AB, you have to have the genotype of IA, IB. That's the only way that you can get both A and B expressed on your cells. If you're type A, you can have IA, or you can have IA little I, right? Both of these will make you only A. It's just that one of them you'll probably make more, and then one of them you'll make less. B, same deal. You can be homozygous dominant. Why did I say A? I meant B. Or you can have heterozygous genotype. And then O has to be recessive for both the alleles. So that's the way that blood type is inherited. And let's see how that plays out if we have two people with different blood types and they have a kid. So if we have somebody with heterozygous type A, so that's this, and somebody who is AB, that's their genotype, right? Well, their kid could be homozygous for A, they could be heterozygous for A, they could be AB, or they could be heterozygous for B. So all the genotypes are different, but if we're looking at the phenotypes, we would have 50% probability of type A, we would have 25% probability of type B, we would have 25% probability of type AB, and we would have 0% probability of O. So that's blood type inheritance, and it's got some pretty interesting applications because if somebody goes and gets a blood transfusion, if they're, let's say they're type A, and type A only, right, not B, and they get type B or type AB blood, their body's going to get mad at that cell surface antigen of B, and it's going to say, that's not me, that's something bad that I have to kill. And so it turns out that type O is the universal donor because it doesn't have any antigens that could possibly upset anybody. And type AB is the universal receiver because there's no antigen that your body won't recognize on the surface of blood cells. There are also some traits that are not on our regular chromosomes. They're actually on our sex chromosomes, X and Y. So females, people who are biologically female have two X chromosomes, and people who are biologically male have an X and a Y, which makes this type of sex-linked inheritance pretty interesting. X-linked is a lot more common than Y-linked. The classic example of X-linked inheritance is hemophilia. Hemophilia is a clotting disorder. So basically, the system that makes your blood clot and your makes you stop bleeding when you get hurt is kind of broken, and so you have to be very careful not to injure yourself if you're a hemophiliac. Hemophilia is recessive, and like I said, X-linked. So let's say that we have a person who has two X chromosomes, and this person is heterozygous, or a carrier for hemophilia. And when we say carrier, all we mean is that this person has an allele for this trait, but does not display it. So this would be like X capital H and X lowercase h. And then we have an XY person, and this person is hemizygous dominant. And we say hemizygous because we only have one possible allele. So let's cross these people and see what happens. So let's put the X chromosomes up here and the XY on this side. And this is going to get us one child who is homozygous dominant, so that child is normal. And we have a carrier, or a heterozygote, also normal phenotype. And then we have our XYs, so this XY is regular. You cannot be an XY carrier because you don't have two alleles to carry something. The only allele you get is going to show. And that's the case here, is that you have hemophilia, you're hemizygous recessive, and this one is affected by hemophilia. So even if neither of the parents shows the trait, one of the kids can get it. And it's going to be likely that this kid's chromosomes are XY because you only need one bad allele in order to show the phenotype. Meanwhile, everybody who's XX is going to be fine in this Punnett square. There are Y-linked disorders and traits. I don't know any off the top of my head, so we'll just make up a Y-linked dominant thing. We're going to call it A. So it's got this allele or this allele. And let's say that we have, well, I mean, XX doesn't matter. And then we have somebody who has, let's say, recessive on the Y chromosome. So this will get us, anybody with XX doesn't care, and anybody who is hemizygous is going to have this trait, whatever it may be. The last type of inheritance I want to touch on pretty briefly, because we don't know that much about the specifics, is polygenic inheritance. And this is basically when more than one gene dictates the phenotype. So for example, skin color, we can't pin down one gene and say this gene dictates our skin color or melanin production. There are many genes that are implicated in this. Other traits are like this. I can't think of any right now, but I know they're out there. But all you have to know about polygenic inheritance is that it's multiple genes making a phenotype. You're never going to be asked to do a Punnett square for a polygenic inheritance thing because they're not simple enough. So I hope that gave you a good idea of genetics and how to predict inheritance patterns.